Okay, the last assignment we did a mitosis storyboard, which you'll recall is the process of taking a diploid cell and making two new diploid cells, which looked a little something like this for a human being. A cell with 46 making two new cells that also have 46. But that explains how to make body cells or somatic cells. What we want to do today is try to make a diagram to demonstrate, well, how do we make the sex cells, the cells that have the half number of the diploid number, which in the case of human beings is 23, being the sperm and the egg. So we're basically going to do two divisions. Instead of doing making two new cells of 46 from one with 46, we're going to do an additional split to where we're going to end up with four cells that all have 23 chromosomes. But in the case of what our little demonstration today, we're going to use a diploid number of 4, which is going to make two new cells that have 4, which are going to be further divided to make haploid cells or sex cells that are going to have 2. And the whole point of this is, if you were to have a sperm with 2 chromosomes, it would meet an egg that has 2 chromosomes, and you would have a new organism that has 4 when those are combined through fertilization. So just as in mitosis, whenever we start out with meiosis, we have to have a period of growth, and a period of growth to get the cell big enough so that it can actually reproduce. And we also said and this is called interphase, just like it was in mitosis. And we said that in interphase, not only is there a period of growth, but we said that the chromosomes are going to unravel into chromatin, or DNA, and make copies of themselves. So I'm going to draw a number 1 for Dad. And there's going to be a copy of his number 1. There's a number 2 for Dad, and a copy. And there will also be a number 1 for Mom that Mom gave to this cell. And there will also be a number 2 that Mom gave to this cell. Of course, at this point, we still have a nucleus. In prophase, this is where things get drastically different from mitosis. In meiosis, there's actually going to be 2 divisions, like I said. So we're going to have prophase 1 to start out with. And what really drastically makes this meiosis different from mitosis is that in prophase 1, Mom's number 1 chromosome and Dad's number 1 chromosome actually find each other. And so I'm going to tighten these back up into chromosomes and draw Mom's number 1. And here's Dad's number 1. And when they find each other, they physically cross over one another and exchange genes. And depending on how random it is, sometimes there could be hundreds of genes interchanged, and sometimes there might not even be 50. It just depends on how much crossing over occurs. It's all completely random. And again, Mom's number 2, there's Dad's number 2 actually, is going to locate Mom's number 2, and they will be crossing over as well. That is the most significant thing that happens. Yes, the DNA tightens back up into chromosomes, but the most significant part of this phase, prophase 1, is that crossing over occurs. And then again, that's an exchange of genetic material. So just like in mitosis, we now move over to what is called metaphase 1. And if you'll recall, in mitosis, the chromosomes line themselves single file down the center of the cell. In this, metaphase 1 of meiosis, we're going to have Mom's number 1 and Dad's number 1 lined up next to each other. So it's not a single file line, it's actually 2 by 2 here. And there's Mom's number 2, and here's Dad's number 2. Now what's really interesting here is these really, even though I have red for Mom and blue for Dad, if you'll recall over here, there was crossing over that occurred. So these really aren't going to be necessarily Mom's and Dad's chromosomes that were given to this cell after all, because on Mom's chromosomes, you're going to have some of Dad's genes that were exchanged. So I'm going to put some blue dots here to show where they had actually exchanged genes. And in those same spots where I put blue on the red, I've got to put red spots on my blue chromosomes to show that those genes were swapped. And just like in mitosis, we start to see spindle fibers showing up. Next, we're going to move on to anaphase 1. In an anaphase 1, our new chromosomes that have been recombined of mom and dad's information are now going to be pulled over to opposite sides of the cell. Now, when mitosis, we were hauling over single chromosomes. We've now got complete pairs that are still being pulled over. I'm going to go ahead and draw that. You can see that these are being pulled over. I'm not going to go ahead and put all the different changed genes on there, but in your assignment, you have to actually keep charting where those exchanged genes were from crossing over. After anaphase 1, we're now going to go to telophase 1. And in telophase 1, just like telophase and mitosis, we're going to see a pinching off. And when that pinching off occurs, we're going to see that we now have in each of these new cells that are being pinched off, we have the resulting chromosomes that were being pulled aside in anaphase. Now here's the problem. We still, in each one of these cells, we still have the diploid number of 4 chromosomes, and our goal is to get the haploid number, which is 2. So we do not want to do another interphase. If we did that, what we would be doing is doubling the DNA or doubling the chromosomes again. So we're going to skip interphase, and we're going to go to what is called prophase 2. And in prophase 2, we now have 2 cells that we're working with. This one and this one. And basically what's going to be done now is we're just going to go ahead and do what amounts to be mitosis for both of these cells. In prophase 2, these guys are going to start making their way towards the center of the cell. And there is no crossing over again. These are already different scrambled up chromosomes. And from prophase 2, we're going to go to metaphase 2. And in metaphase 2, we now have 2 cells where we have these chromosomes aligned in the center. We also have the appearance of spindle fibers are starting to show up. And then we're going to move into anaphase 2. And in anaphase 2, the centromeres are going to split, and we're going to be sending individual chromosomes to opposite sides of these cells. Again, the spindle fibers are complete at this point. Oops, we're going to have to do that there. The last thing that's going to happen, as you can see, what we need to do here is we need to get both of these cells to split. And when we do that, we're going to call this telophase 2. And we're going to end up with four new cells that all have 2 chromosomes in them, or the haploid number is a better term for it. And if it's a male, these will be sperm cells. And if it's a female, we're only going to get 1 good viable egg and 3 polar bodies, which in other words, these eggs are not big enough to actually do anything. The egg is so big, there's not enough material to make 4 good eggs. So instead, it's 1 good egg and 3 polar bodies, which are no good eggs that get dissolved. So this is meiosis.